as we all know that the pharma industry, the R&D spend is around $160 to $170 billion per annum. And out of this, around 20 to 25 billion is the discovery outsourcing market, which is quite a large chunk. And there are quite a few service providers looking at this opportunity, as the drug discovery services is a very vast landscape with both small and large CROs and companies, other companies vying for this opportunity close to 20 to 20 billion. Some of them have focused on chemistry, that is, some have maybe 100, 2,000, 3,000 FTs, chemistry FTs in the synthetic area, while others have focused on maybe specialized areas of pharmacology, that is, they offer pharmacology in the different disease areas in oncology, inflammation, pain, metabolic disorders, etc. And also over the last 3 to 4 years, they have added some newer, that is patient-derived cell lines models as well to improve on their translational to the clinic. And some companies are focusing on toxicology, and some of them are only focused on the DMPK alone. That is, they have DMPK services starting from early discovery to late discovery and ending up into the preclinical discovery as well. So the overall market, which is, as I said, around 20 to 22 billion, okay, comprises of around 30% is close to it is biology, around 30 to 32%, in which protein sciences and in vitro services, and as we all know that there are a lot of companies offering in vitro assay services, is around 30 to 32% of 22 billion is of in chemistry, in biology. But the rest, that is close to 70% of the market of around 22 billion, which is close to 14 billion, is mostly chemistry and drug discovery services related, that is medchem related opportunities and lead optimized related opportunities, and which is a vast sum of $14 billion. And there are many companies, as I told before, that who are offering niche services, select services, standalone services, that is chemistry, pharmacology, toxicology, or DMPK alone, or in vitro services. But overall, if you look at all the services offerings which the companies are offering, that CROs are offering, there are very few companies and organizations, that is, or CROs, which can unite and connect all these dots. That's the medchem, that the in vitro assays, that the early pharmacology, toxicology, DMPK, to give you an integrated discovery programs. Obviously, the big pharmas and the biotechs have been doing this, but as a CRO companies, there are very few companies all around the world which have the ability to do this. But we at APSL have been doing this in the form of integrated drug discovery programs over the many years. And as we share the core discovery values of the biotechs and the big pharma companies, and we have the expertise and the experts in the subject matter, who can connect and take these programs to final conclusion to give clinical candidates. So with this little bit of background, I'll just go over to the services offerings, the drug discovery services offerings from APSL. Before that, I'll just give a very brief background about APSL itself. APSL is an integrated end-to-end service provider across 3 verticals. That is the discovery services, which comprises of both chemistry and biology. Then there is a development vertical, and finally is the commercial manufacturing. And we are service providers for innovators in pharma, crop sciences, and animal health and other industries as well. We have world-class infrastructure, and we have 2 locations. One is in Bangalore, around 100,000 square feet, and another campus at Hyderabad, which is around 210,000 square feet of lab space. And we have state-of-the-art infrastructure and an environment which is conducive to scientific innovation. And across this discovery and development chain, we have around close to 650 scientists, out of which in the discovery services, we have around 375 scientists in chemistry and biology. And the development has DS and DP offerings, around 200 scientists. And the commercial manufacturing space, we have around 100 people as well to offer these services. Our range of services starts with early discovery to late discovery, and finally to preclinical and clinical discovery as well. So our expertise over the years has been in chemistry and small molecules and peptides, where we can support early discovery in assay biochemistry, cell and molecular biology, ADME, PK, and in vitro tox. And then if you programs moved on to the late stage, that is the lead optimization studies, then we keep adding ADME, PK, in vivo efficacy, and in vivo tox as well. And then as the program moves to a clinical candidate stage, that is candidate nomination, we offer PK/PD services, preclinical services, pharmaceutical development to look at the BAs of the compound. We have both GLP and non-GLP tox. And then once the candidate is declared, we can offer support at both the CMC level in non-GMP and the GMP level as well. That is the DS and DP, both DS and DP in non-GMP and GMP to support efficacy and clinical support as well. So overall, we can support most of the programs, any of the programs, starting from early discovery, that is hit to lead, to late stage discovery, that is lead optimization, getting to a preclinical candidate, and helping you file the IND nomination and candidate nominations with DSTP support to support it in the clinical trials as well. And now that was the overall perspective for APSL. But now moving on to the division that is the Aurigene Discovery Services. So as the name implies, this supports discovery in chemistry and biology for innovators in pharma, biotechs, crop sciences, and animal health. And we have a specialized platform in the peptides. We have around 30 to 35 peptide scientists. And the specialized platform is in the range of the microwave-assisted peptide synthesis, which we have added recently. As I said, we have the state-of-the-art infrastructure and environment, which is conducive to innovation as well. We have, in this area, we have around 375 scientists across the drug discovery value chain with 60 PhDs. And now this 375 is moving on to now close to 400 scientists. And the divisions are the chemistry, which is the largest division, which has around 275 scientists in small molecules and peptides, and analytical division, which has around 30 scientists, and the toxicology, pharmacology, in vitro sciences has around 40 scientists, and the DMPK, that is the part which is both in vitro and in vivo, is around 30 scientists. So both, and these services are provided from both locations, that is Hyderabad and Bangalore itself. So it's common to both. So each site is independent of each other, can support each other. And what are our discovery services offering? We, over the last 10 to 15 years, we have been building long-term partnerships and customized services for our pharma partners and collaborators in the biotech industry as well, and animal health and crop science innovators. Our offerings include standalone medicinal chemistry services in the areas of small molecules, peptides, carbohydrates, and nucleoside chemistry. And we also offer a non-GMP scale-up for efficacy and tox studies. The GMP part is taken up by the development team, that is the DSTP part. And then one of the most interesting and the valued services which we offer is from the integrated discovery domain. That is in the areas expertise in the small molecule and peptide areas, where we do end-to-end discovery. I'll show you an example later in the program, later in the webinar. And we also offer a lot of mix and match programs, that is not fully integrated discovery programs, but integrate sort of a mix and match program according to our collaborators' needs. That is, we do chemistry, medchem plus DMPK, where the other in vitro assays is done by the partner, or we do chemistry and in vitro, but other the model, maybe the pharmacology models are done by our partners, collaborators itself. There are other services that we do chemistry plus scale up, plus chemistry plus DMPK and scale up to take up your compounds to the top. So there are various different mix and match programs which we are running over here at Aurigene. And then to support all the integrated discovery programs, we have the in vitro which has biochemistry and cell and molecular biology teams to support any in vitro assays. And then ADME-PK is a huge department, around 40 scientists, where we support from early to late and to preclinical discovery. We run hundreds and hundreds of assays, starting from solubilities, both in what you can say kinetic or thermodynamic permeability in different assays, that is maybe MDCK, CACO, as depends on the partner, CYP inhibitions, the stability studies, both in microsomes and in hepatocytes. Then we also have an animal house in-house, and it's a GLP certified. We have both a rodent facility and a dog facility to look at the PKs of your compounds, and then we can help in the formulations of your compound as well if you want to improve the overall viability of your compound. And the other department is we have a very good pharmacology department, which has around expertise in different therapeutic areas, that is oncology, inflammation, infectious diseases, and metabolic diseases as well. But if something is not there, we can set it up much easily as well. And finally is the tox department, where we can do both GLP and non-GLP studies as well for the compound to help you take the compounds to clinic and filing the IND nominations as well. And so what is the space we are operating on in the discovery services? As anybody who's done discovery knows that you start off with a target, and the program moves from the hit design to hit to lead and lead optimization to preclinical development, where you end up with a clinical candidate, and then once it is done, then you go ahead and file your IND. So once the target is nominated, we have ability to look at the hits from various areas. It could be a fast follower program, or it could be a virtual hit, or it could be a screening effort, or it could be an SPDD-led effort. So the hits could come from different areas. Then we look at the hits, could either be from 1 to 10 micromolar. We look at the hits. Is there an opportunity, FT opportunity for these hits as well? Then as the program moves forward, we work on at least 2 to 3 different series, and the compound starts to become more potent, that is become less than 100 nanomolar. We look at the SAR of these compounds, we establish the mechanism of actions, and also look at the primary efficacy, that is of the compound. Mechanism of action means we will set up the biochemistry and the biochemical in the cell-based assays for these compounds. And once the compound moves to little more in the lead optimization stage, the compounds do start to become much more potent, that is less than 50 nanomolar as the TPP defines it. Then also is by the side of SAR, that is structure activity relationship. We also start to look at the structure property relationship of these compounds, and the properties also start to become much more important as the compound need to go to the development stage. That is, the permeability and the solubilities also need to be defined so that the compounds could be developed. And we look at the PK/PD of these compounds in the ADMEs, and we start to look at the PKs in the higher species as well. And we look at efficacy of these compounds in different models as well. And once the compound reaches the preclinical development stage, we look at the development candidate, could be 1 or 2 compounds or 3 compounds, then try to choose which is the best one, is it? And if there is some lacunas, we see the formulation department could help. Then based on the toxicity at the ADME profile, try to choose one clinical candidate that is just taken forward by the IND to the IND nomination. And so how this discovery is done is usually led by a chemistry and a biology coordinator. And the teams are formed from different departments, starting from biochemistry and medicinal chemistry. Medicinal chemistry includes both medicinal chemistry and computational chemistry, plus the synthetic chemistry aspects as well. So plus analytical chemistry to support the synthesis. Then the cell and molecular biology departments look at it, and ADME, PK, pharmacology also chips in. Then the pharmaceutical development comes late stage in the program, and the safety and the toxicology also comes up in the late stage of the program. And finally, once the candidate is done, the scale up and the process teams also come in to look at the most efficient routes of the synthesis. So at each stage of the program, that is hit to lead or lead optimization or preclinical development, a target product profile, that is a TPP is defined at each stage and each criteria is met to move the compound to the next stage, and a program flowchart is mapped, which I'll show you in the next slide, that how these programs are run. I'll show you a typical flowchart, how the programs are run over here. The next slide shows what is the program flowchart and shows that the different departments which are there, starting from biochemistry, cell and molecular biology, DMPK, pharmacology, safety, and tox. So it all starts off with the design and the synthesis. In the early stage, the compounds are designed by iterative process and synthesized in very small scales of 20 to 50 milligrams, then subjected to the biochemical assay that could be an enzyme assay or could be a binding assay. And we look at the selectivity parameters of the critical, suppose it's a kinase program, we look at the critical kinases, 2-point screening. So once it passes through that hub of around 50 nanomolar, then we start to looking at the cell-based assays and the mechanistic assay and both the functional assay. Again, there is a criteria for this, that is 100 nanomolar, because then we also start to look at its early admin properties, that is the solubilities, could be a thermodynamic solubility. The criteria is around 20 micromolar, or we look at the microsome or the human-like mice or the human liver microsome stability of these compounds, so that they could go into the PK in the next step. These compounds are stable. Then we also look at the permeability. So if these passes these parameters of around 100 nanomolar, then we move the compounds to the mice PKs, and we look at both the IV and the oral route. Then we defined a little bit of profile that it could, the 20% could be the BA we are looking for. And if these are not met, then we look at the reason that could be the CACO permeability. We look at the flux ratios, we look at the CYP inhibition, the PP, the amount. Then also start to look at the hERG of these compounds. There are 1 or 2 series which will help us define that is which series to take forward if any hERG liabilities are there in any of these series. And then the compounds are scaled up. If you look at the PKs are fine, the permeability is fine, everything is okay. Then we start the scale up compound a little bit to around 500 mg to look at the PK/PD in mice. And then look at the dose escalation studies, that is both in mice and rats, around 10, 30, or 100 MPK. To look at it, if the dose proportionality is there, or is it linear, or is it just dose proportional. Then look at the early MTD, that is the maximum tolerated dose in the mice. Then also start to evaluate of the physical form, that is either it is amorphous or crystalline, or assessment of the chemical and thermal stabilities and different pHs, acidic and basic pHs as well. Then once the PK/PD is done, if it matches the PK/PD, if the compound matches, we take the compounds again scaled up a little bit more to 10 grams to 100 grams, depending what could be done by the medicinal chemistry team or the process development team as well, because if it is reaching the lead optimization stage, the process development team also starts to come into the play. And once the compound is scaled up, we look at the efficacy of the compounds, and efficacy is matching. Then we look at the in vitro safety, again, the hERGs and the rat, the cardiovascular and the mini-AIMs. Then we look at the cellular selective, that is, if the compound is in oncology, then we look at it, it is only hitting the cells, only hitting the cells it is desired, and is not hitting the normal cells as well. Then we look at the 14-day non-GLP tox studies, the DOT PKs, the hepatocyte stability, the plasma stability, the CYP TDIs, that is, and we kinome and receptor panels also look at the compound is safe and only hits the desired protein which leads to the nomination of a candidate. So this is a brief, what you can say is a program flowchart. That is how we run our discovery program. That this was something about from the lead optimization stage leading to a candidate nomination. And next, I'll just show you an example of this integrated drug discovery program, which we had done some time back and was published in the year 2014 in JMedChem paper. So this program was lasted around 3 years. It was a fast follower program. and we published two patents out of this. And as I had entailed before, that at each stage we try to follow two series. That is, one is the lead series followed by a backup series for the compound. And in this case, this is the fast follower design philosophy was we do scaffold morphing. We started off with a 5,5 hydantoin scaffold and ended up one of the series was the 5,5 pyrazolene ring. And we saw the crystal structure of the compound, which is shown over here in this. And the crystal structure was also solved in-house for this compound. And the route of synthesis is shown. It's quite an easy and the regioselective and the stereoselective synthesis. And starting off with the proline, derivatized proline, hydroxyproline, and which was cyclized with the hydrazine to give you the right compounds. And the small table on the right shows the SAR of this compound. The comparator was a dihydroxytestosterone, which was quite potent for it. And the assays was done in the C12 cell line, which is the mouse muscle cell lines. These are the mouse muscle cell lines. And one of the criteria was that this compound 26, which was chosen as the lead compound, as nominated as a clinical candidate, the approach was that though this compound was orally bioavailable, the aim of the project was it was to be given through a patch clamp, and the criteria was met. And you can look at it, which is the assay shown that it was quite permeable through the patch. It was because it was highly soluble and highly permeable to the patch. And on the right hand side, the compound 26, what I have done is I've shown the efficacy data for this compound. The efficacy data is shown as in the Hirschberger in vivo assay. And the two muscles which it was acting on was the prostate and the levator ani. Levator ani is your hand muscles, and the prostate was the undesired muscle. So the effect of the compound usually is that it will help strengthen the muscles when people become aged. So it will usually increase the weight of the muscle. So that's how the Hirschberger assays are done. And if you look at it at the lower doses, there's a very nice separation between the levator ani and the prostate. So that it was really strengthening the muscles and increasing the weight of the muscles at the lower doses. But unfortunately, at the higher doses, the gap narrowed down. So as this compound was nominated, but unfortunately, it was not taken up to the clinic, because it was thought that at this stage, when we give higher doses, the tox window is not there. So that's the theory that this program was stopped at this stage. But this is just an example of how the programs are run. That is, we start off with chemistry, medchem, designing, synthesis, set up the assays, and do the efficacies and the safety studies as well to come up to clinical candidates. In the next slide, so this is a program for integrated discovery, that is end-to-end discovery. I'll take you very briefly also about the mix and match programs which have been doing now. And one of the examples I've taken is also published in the GMedChem, and it is shown here on the left. The mix and match services are that we offer medicinal chemistry plus DMPK. We offer medicinal chemistry, is not synthetic medicinal chemistry, but medchem design with the help of computational, and then do the DMPK part, that is early discovery, the early DMPK or late DMPK leading to a clinical candidate. And most of the biology, that is in vitro assays or the efficacy models, are done at our collaborator site. And then the other offerings which we also have is the medchem design and synthesis, where we design, iterate, synthesize, develop SAR, and give it back to the partner. Other services are chemistry plus DMPK plus scale up plus tox services as well. So once the candidate is desired, we take it up either for scale it up for efficacy and then for tox as well. And the fourth services which we also offer and also running is chemistry plus in vitro. That is, we only do the chemistry plus and the assay part. We both do chemistry and in vitro, both in biochemistry and cell-based assays. And all of these services, all of these, what I talked about, that integrated discovery services or the mix and match services, are helped by different departments. And small, as I told earlier, that the small molecule chemistry department is one of the largest departments. We have around 275 scientists around it, and close to now adding another 25, 30, so it will be close to 300 scientists. And we offer all types of chemistry, starting from sensitive reactions from gram to kilogram, chiral chemistry, hydrogenation, LDA-based, boron reduction, other types, COP. But what I want to focus is that we usually have expertise in different areas of chemistry as well. That is, if you look at it, I've shown an example of the highlighted one of the covalent kinase chemistry example, which is the JMC paper, which I showed in the previous slide. So that was a clinical candidate. So the program was done here. So this is the covalent kinase chemistry. We have numerous examples from carbohydrate chemistry as well, which I have not shown. But we, over the last 3-4 years, we have also developed expertise in degrader chemistry, which is, as you know, 2 programs now from Arvinus has moved to clinic, that is in the androgen receptor areas and the ER area. So we have, over the few years, we have expertise in both degrader chemistry and biology as well. And recently, and we can support any of the degrader program for any of our collaborators. And recently, we have added some new niche chemistry technologies as well. What I have shown is a photoredox catalysis reactions. We have 2 systems of it. I have shown an example of it, of this type of chemistry, this type of chemistry as well. And that we can do, this is much cleaner, quicker, faster chemistry, which can help solve many of these chemistry issues, give you much cleaner reactions and much better yields. So over the years, we have developed expertise, an excellent track record in challenging chemistry, in multigram specific and focused libraries as well, in different areas, that is covalent chemistry, degrader chemistry, kinase chemistry, and different, you can say NHRs, different kind of chemistries as well, for help supporting our collaborators. And so to support our chemistry programs, so what we have also done over the last 5 to 6 years, we have collected an inventory of around 50,000 chemicals on, in-house, and which will help jumpstart your chemistry synthesis, both in chemistry synthesis and medchem program as well. This is searchable, and it is located as in one location, and it is either searchable by name, CAS number, or structure, substructure search. And 50,000 and 10,000 of these are in the cold storage. I've just shown an example where it has helped us kickstart some of the quicker chemistry. Suppose there was the indinone was known to us, and our collaborator wanted to do different chemistries. One was the reductive amination, and one was Suzuki coupling. So as we looked up to the inventory, we gave them around 1,000 boronic acids, which were available off the shelf to us, and another 500 different amines, chiral amines, which were available off the shelf to us. So we suggested both of them to our collaborator, and the collaborator chose 10 of each from them, and we could quickly start doing both the chemistries in a single day's time. So this is how we can jumpstart most of your chemistry issues and help you solve chemistry problems as well. And as I indicated earlier, this slide shows our peptide synthesis expertise. So we have been, I said, we have around 30 to 35 peptide chemists with expertise based in solid phase and solution phase techniques. And recently we've added a SEM, automated microwave peptide synthesizer, which can synthesize compounds up to 5 micromolar scale, 5 to 10 micromolar scale, and help reactions which were earlier used to take days, around 1 to 3 days, that is the amide coupling reactions, can be finished in less than 15 to 20 minutes or less than an hour. And to support all of our chemistry efforts, we have 3 lyophilizers, 3 analytical HPLCs, and PERP HPLCs with Agilent and Shimadzu, and LC-Mass single quad mass spectrometer. We have standalone facility for the peptides, and our expertise ranges from in different areas of chemistry as well. As you can see, the lactams, cysteine couplings, pegylated peptides, and all kinds of different peptides are there. We have the expertise to help programs either in integrated discovery form or in just FT-based chemistry services as well. And to support each of our integrated discovery efforts, it is important to have all the equal teams. So we have a team from the biology part which sets up the biochemical assays and the cell-based assays. Both in the biochemical assays, we have run programs in kinases, NHRs, GPCRs, phosphatases, degraders, as I said, and epigenetics as well, both in the reader class and the writer class as well, proteases and the dehydrogenases. And the technologies which we have using biochemical assays are absorbance, luminescence, fluorescence polarizations, and fluorescence energy, that is the TR-FRET assays as well. And in the areas of cellular assays, we do target modulation, functional activity, look at the predictive efficacy, the MOAs, if the kinase target, or the potential toxicity of the compound in different cell lines, to make sure the compound is selective and only acts on the desired cell line and not on the normal cell line as well, and look at the biomarkers as well to take the compound to the clinic. So we have expertise as standalone services or as an integrated or a mix and match services, starting with enzyme inhibition, binding assays, or cellular assays can be developed as per your target needs. Then one of the important parts for the discovery is the DMPK team, which supports both the discovery and the development part of the programs. At the early discovery stage, we look at the physicochemical and the druggable properties of the compounds. We measure the solubilities, be it solubility could be the thermodynamic or the kinetic property, look at the log D, look at the chemical and the metabolic stability of the compounds in different liver microsomes, that is human, mouse, rat, to look at, and then look at the permeability of the compounds. If compounds are not permeable, we can look at the flux ratios as well. In CACO and MDCK, we look at the protein binding and the CYP inhibition. And as the compound moves forward to the development stage, then we look at the optimization of the drug properties. We look at the PK/PD, the IV-IV correlation. We look at the drug-drug interaction that between the CYPs. We look at the target issue exposure. We look at the blood plasma ratios, and on the tissue distribution, the compound is distribution to the right tissue, and we look at the PK/PD of the compound, and the med-ID, and once we have all these, we can also have a look at the first in human dose prediction, that then we have the ability to do that. And once the clinical candidate is declared, we come to the regulatory aspects of these compounds, that we help the IND and the AND studies in the regulatory. We can help in the DMPK package of the INDs with the test and the reference formulation assessment. And we look at the biowaivers in single, multiple dose, and the tissue distribution studies for the compounds as well. So it's an overall package. It is not that stops at discovery. We have DMPK services, integrated DMPK services from discovery, development, and regulatory as well. And we have a very strong in vivo pharmacology department to support any of the efficacy services which are needed to run programs to lead to a conclusion. And in the oncology area, we have set up syngenic models and the xenograph model. And this is one of the larger areas. And we have models in the metabolic disorders, both in diabetes and obesity, cardiovascular, and atherosclerosis. And in the areas of, we've run programs in pain and inflammation. We have neuropathic pain, visceral pain, and other inflammations, arthritis models, different dermatitis models, colitis, asthma, and multiple sclerosis models as well. And over the years, we have also run programs in the areas of anti-infective, that is antibacterial and antifungal. So we have both systemic and organ bundle models. And if some of these models are not there, in the areas of therapeutic are not there, we can develop models as and when needed, like we have done, which are shown here. That is the muscle injury and the regeneration model. So the expertise to set up any of the models is existent by the team over here, and can be taken up with a 2 to 3 months timeline to set up the efficacy models. And one of the last areas is the toxicology department. We have both GLP and non-GLP, and it's one of the most important areas which makes or breaks the program. That is, the compound is toxic, it's very difficult to take it to the next step, that is, take it to the clinic. To help support the programs, and also as a standalone services, we have a built for purpose and standalone state-of-the-art toxicology and animal house spread over around 18,000 square feet, where we do different kind of assays to support your drug safety and therapeutic calculation of the therapeutic index for your compound. We look at the in vitro mutagenicity, that we have the Ames test, and the chromosome aberration test as well, micronucleus test using human peripheral body lymphocytes. And we also look at the in vivo mutagenicity of these compounds, and the cardiovascular safety pharmacology in the case of HERT binding or automated clamp assay. So for the HERT binding, we have the dofetilide assay, and we have a patch clamp, a manual automated patch clamp. We also look at the impurity qualification for your compounds in the rats and mice to help measure the level of the design impurities. And as a general toxicology studies, we do acute toxicology studies in rats and mice, both in GLP and non-GLP. And we do non-GLP exploratory dose range finding study, that is MTD, in rats and mice, subacute studies starting with 14 to 28 days, and leading, if it's a inflammation program, then we need to also do chronic studies, that is starting with for 90 days, and the chronic toxicity from 6 to 12 months as well, as the case in rats and mice. And we have a clinical pathology and histopathology certified people who can take up the pathology and the histopathology of the compound. And to support all this, we have analytical and bioanalytical support to support both the GLP and the non-GLP tox studies for the compound, and an independent QA to manage the whole system, and data management and archives to do this. And over the years, last few, and one more services which we also offer is the pre-formulation services, which is also at the end of the program. Once you have 1 or 2 compounds and want to look at improve the properties of the compounds, not by chemistry, but by looking at the formulation services, that if you can improve the oral viability of the compound, or you can make the compound a little more permeable. So we can look at the biopharmaceutics of the compound, the solubilities. We look at different pHs and the dissolution of the compounds, and you help enabling the preclinical formulation of the compound in different ways, either by solid dispersions or lipid-based complexation or nano suspension as well. We have the ability to do parental formulation, and then look at the GL and take it to the GLP formulation as well. And once you come, we have an analytical team to support all this, to all these studies, both with the help of the assay and the stability studies as well. And comes to the preclinical formulation studies, we can look at the in vitro phototoxicity of the compound, this molar absorptivity, solid state characterization of the compound, and look at crystalline or amorphous. We look at the bulk properties, polymorphs of the compound, hygroscopicity, and API stability at different conditions are there. We can do it both GLP, non-GLP conditions, and come to the last part, that is the drug substance. And all of this, as I told about the toxicology and the both GLP studies, we have a very good track record of regulatory inspections, and track record is pretty clean. If you look at the last part, we have been certified by the NGCMA India as well as the SPSA Netherlands. And over the last 3 years, starting from 2016, 2017, 2018, and 2019 as well, We have been audited by the US FDA, and all the time we have got no 483s, and the facility is really good to support a lot of the GLP studies as well. So this is the brief overall of what overall services we have to offer in the areas of drug discovery. And just before ending this, I just wanted to highlight a few of the Aurigene advantages. That is, we have an experienced and a high quality scientific and a scale-up team that is end-to-end discovery, taking up their compounds to clinic as well, which is well versed in the areas of discovery and CMC services. And we have a strong and sustainable operations supporting long-term partnership. That is what we are looking for. And we have a dedicated manufacturing unit with pilot and commercial scale capabilities. Suppose the compound passes phase 3 and you want to take it to commercial, we have the capabilities to do that. And we hope we are a partner of choice for discovery, clinical and commercial phase for your compounds. And we have different flexible business models to support any of the services, and flexibility to partner across academics, biotech, startups, large pharma companies, or animal health, or other areas as well. So we focus on creating long-term associations with quality science, and over a period of time, we want to be a contributor to our partners' development profile and help them commercialize their products and take it up. And overall, what I can say is we offer services from protein sciences, from chemistry, medchem to DSTP development and manufacturing services overall. And overall, and the summary and the recap is that we have over the last 10 to 15 years, we have small and large FT discovery and services collaborations in peptides, small molecules, DMPK, pharmacology and toxicology, integrated services, and mix and match services as well. We offer end-to-end services, starting with chemistry synthesis to lead optimization, followed by preclinical safety and evaluation, and regulatory tox services as well, and the formulation department. We have a technology-led services with a wide range of capabilities and GMP infrastructure, and ability to flexibility in manufacturing, assurance of supplies, and regulate and EHS controls are there. We can support both early and late phase drug substance and drug product clinical supplies, plus, as I mentioned earlier, large scale commercial supplies with complete CMC support to enable your discovery programs, both in the early discovery, late stage discovery, and as well as the clinical support followed by commercial manufacturing as well, to help support take your compounds to clinic and to market as well. And that is all we have for today. And thank you for your patience.